Our lecture topic today is inflammation and the inflammatory process. The focus or the outcomes will include taking a look at the physiology of the inflammatory response itself, which will include the vascular, the plasma protein, and the cell mediator responses and how they contribute to that inflammatory response. We're going to talk a fair amount about cytokines, both physiologically as well as pathophysiologically, and how they work in the inflammatory process. It's important that you have an understanding of the pro and the inflammatory cytokines, interferons, and tumor necrosis factor. And those are some components that I would expect that you'll have a good understanding of and be able to identify how they play within that process. We'll talk a lot about mast cells and also about the role of histamine in the inflammatory response. Can't talk about the inflammatory response and inflammation unless we look at phagocytes, especially the neutrophils, eosinophils, basophils, monophytes, and the NKs, or the natural killer cells, and then talk about how that process of phagocytosis actually occurs. And then we'll end our time together talking about the manifestations of acute inflammation, try to pull everything together. So when we think about inflammation, and we talk a lot about inflammation in our daily lives as well as in our professional lives, but we have to remember that inflammation as a process is triggered when that first line physical, chemical, or mechanical barrier has been breached. So when we think about the physical barrier that's been breached, we think about skin, the epithelium. The chemical barrier would include the mucous membranes, because we know that mucus is very useful in both trapping as well as assisting in killing organisms as well as particles. And then the mechanical barrier, sometimes we don't think much about mechanical barriers, but if you think about vomiting, urination, diarrhea, those are all barriers that we have to help us remove substances that don't belong or that we don't believe belong in the body. We also know that the inflammatory response is rapid, but it's non-specific. And what that means is it also has no memory because it's not specific. So as a result then, we know that that rapid inflammatory response, which is non-specific, we can actually measure that by using what we would call the inflammatory markers. Probably the two most common inflammatory markers you're aware of include the sedimentation rate and C-reactive protein. If you've worked with either one of these markers, these blood tests, you know that they tell you that there's an inflammatory process going on, but it doesn't tell you where the process is occurring. So that's what we mean by it being non-specific. Basically, it gives you a license to hunt, to figure it out. So as we talk about inflammation, we'll talk about three parts of the inflammatory response. We'll talk about the vascular response, the plasma proteins response, and the cell-mediated or cell mediator, as well as the activity of cytokines and the response that would bring. So let's start with the vascular response. First and foremost, remember that all of this starts with injury and the activity of the mast cells. There is a great figure in your textbook on page 206, and especially for those of you that are visual, take a look at that figure. It really is the book, I can't say enough about the great figures and tables that they include to help us with our learning. Remember that from your basic patho that mast cells are a type of white blood cells, and they contain granules. These granules have histamine, leukotrienes, and prostaglandins in them. Therefore, when a mast cell degranulates, the histamine, the leukotrienes, the prostaglandins are released into the circulation and released into the area of inflammation. That's an important concept to remember. As we look at the vascular response, we look for those signs of inflammation: redness, heat, swelling, discomfort. And those signs of inflammation are due to changes in the small vascular structures. Now, a lot of these signs of inflammation are due to the response to histamine. Remember that histamine causes capillary vasodilation as well as increased permeability of the cell walls and the cell membranes. Inflammation, the redness, the heat, and the swelling also is under the influence of the leukotrienes. Leukotrienes also increase permeability as well as assist with leukocytes concentrating at the area. Now remember that how we respond to histamine depends on the receptor to which it binds. There are H1 receptors and H2 receptors. H1 receptors are what we might call the pro-inflammatory or inflammation type receptors, and these are the ones that are in smooth muscle cells. We think of them being within the lungs. H2 receptors help to dampen that inflammatory response. And a lot of our H2 receptors are actually in the GI tract. So we have vasodilation, we have increased permeability, and we have movement of our white cells to and through the vessels. So that's a very important part of this vascular response, remembering once again that the vasodilation and the increased permeability is under the influence of histamine and leukotrienes, and that facilitates the movement of the white blood cells. The key to the rest of the response is to deliver other response materials as well as remove wastes from the area. So we want to be sure that nutrients and oxygen are coming to that area. Vasodilation, permeability help with that, but also have to remove waste products and cellular debris. So there has to be a housekeeping function as part of this inflammatory response as well. The other piece we know is that when we have the vasodilation, the increased permeability, and the movement of white cells to and through, these are associated with the release of cytokines, especially TNF or tumor necrosis factor. This vascular response plays an incredibly important role not only in the inflammatory responses, which is our innate and natural immunity, but also in our adaptive immunity, in making sure that the B and the T lymphocytes get to where they belong via as well as using the lymphatics to clear the area of debris. The second part of the inflammatory response are the plasma proteins. When we think about plasma proteins and the inflammatory response, we're especially thinking about complement, the coagulation factors, and the kinins. So let's look at each individual one of these. Complement can cause either direct destruction or, as many of these things we're going to talk about in this response, activates other components. Now, the classical pathway with complement is an antigen-antibody complement complex. Remember, antibodies are protein structures, so there's a complex that actually is formed as a result of this plasma protein response. When you activate the first step, that then leads to a sequential activation of other components. And by activating all of these components, you end by forming a membrane attack complex, which then ends up resulting in cell lysis, which was the goal of the inflammatory response. Complement also further facilitates mast cell degranulation, which, as we know, leads to initial steps. Leukocyte chemotaxis, which is getting to the area of the injury, the inflammation. Opsonization, which is recognition and adhesion, and then lysis, which would be the killing. So opsonization becomes a very critical part then of what happens in this plasma protein response. It's one of the steps, because with opsonization it actually allows antibody complexes to make bacteria more susceptible to phagocytosis, and it does that by binding on the phagocyte surface. So again, stepwise activation. The next piece of the plasma protein response are the clotting factors. Now, the clotting factors are initiated and are stimulated to prevent spread of this whatever the offending thing that causes the cell injury. The clotting factors are there to prevent the spread of this offender to nearby tissues, to trap invaders, to achieve hemostasis, and then also begin laying down that fibrin meshwork that helps provide repair and healing of the organism. And there are numerous stimuli that actually will encourage those clotting factors to be produced, both protective as well as some of these stimuli for the clotting factors actually come from the invader itself. Therefore, these clotting factors are have a highly protective function. They help us build the fibrin mesh as well as assist in containing the offending stimuli. The last part of the plasma protein response are the kinins, and the most common kinin that we usually have is bradykinin. Bradykinin has a lot of local effects and primarily works through its local effects to achieve vasodilation, encourage vascular permeability, smooth muscle contraction, nerve cell stimulation, and once again, leukocyte chemotaxis. So you can see that plasma protein response doesn't just do one thing, it's doing a number of different things, and that's what the kinins are responsible for. Kinins are found both in tissue as well as in body fluids, but the thing we have to remember about kinins, and it's important in this mechanism, is that they are short-acting because they are rapidly degraded. So you're constantly having to replace those plasma proteins. The third part of the inflammatory response is the cell mediator response. We're going to talk about cytokines in the context of the cell mediator response. A lot of the cell mediator response is activated by our plasma protein systems like complement, the coagulating or clotting factors, and the kinins, as well as during cell destruction itself. So that cell mediator response not only is activated by that second component of the plasma protein response, but also cell destruction itself. In that cell mediator response, a ligand binds to cell receptors. Now, a ligand is a molecule that binds to a receptor and forms then a larger complex. And this ligand binding can be to a very small or a very large variety of antigens. As that ligand binds, you have that ligand binding, it activates intracellular signals and then further activates additional cells. What's really interesting about the cell mediator response also is that these receptors may recognize patterns. They may recognize products of cell damage. They may recognize the complement has been involved. So as a result then, in this cell mediator response, receptors have a wide recognition ability. And one would presume that this is probably because there's a very large variety of potential antigens that our body has to have the ability to respond to. So what are these cytokines that we keep talking about? Well, cytokines, very simply, are soluble factors that are actually secreted by cells, and they contribute to our resistance because of their specific receptor binding ability. Cytokines come in two flavors. They're pro-inflammatory or they're anti-inflammatory. Once again, like those kinins, cytokines tend to act locally. They bind to targets, and then they have the ability, as a result of that binding, to alter the target cell's function. And that's very important. Now, although they act locally, many of the effects that we see from a cytokine are systemic. So, for example, fever is caused by cytokines, specifically IL-1. Now, The more cytokines you have being produced, the greater the response. And that makes sense. So it's an order of magnitude. There's a very interesting concept and theory that has been gaining traction over the last couple of years. And I will tell you, this is not in your textbook. However, this is an important concept that I think that you need to be aware of. And this is the concept of inflammaging. And inflammaging recognizes that these cytokines can be potentially damaging and provides a theoretical link between chronic disease and chronic inflammation. The idea behind inflammaging and the physiologic theory of inflammaging is that chronic inflammation actually damages cells and can work to weaken our immune system. We know that chronic inflammation has been linked to heart disease, to Alzheimer's, to insulin resistance, and definitely in frailty, especially in elders. It is also proposed that we can protect ourselves from this inflammaging or this chronic inflammation. And we can do that through a diet high in phytochemicals, which is your plant-based diet, and antioxidants. And antioxidants like vitamin E. So what are some of these cytokines? Well, let's talk about some of the three most common groups: interleukins, interferons, and tumor necrosis factor alpha. Interleukins are messengers, and these messengers are produced by macrophages and by leukocytes specifically to assist in enhancing the adaptive immune response. So there's an important role in initial inflammation as well as a longer-term role in our adaptive immune response. So we have pro-inflammatory and we have anti-inflammatory interleukins, messengers. The two most common, so I'll just give you a couple of examples. IL-1 and IL-6 are pro-inflammatory cytokines. IL-1 is produced by the macrophages, and you'll see the presence of IL-1 in times of infection. IL-6 is produced by macrophages, by lymphocytes, and by fibroblasts. Now what IL-6 does, though, is it actually induces the liver to produce proteins that we need for inflammation. So remember, we talked about some of those plasma proteins that we would need. The most common anti-inflammatory cytokine that we think about is IL-10. IL-10 is produced by the lymphocytes. It has the ability, interestingly enough, to actually suppress lymphocytes, but most importantly has that ability to suppress IL-1 and IL-6. Remember, those are the pro-inflammatory cytokines. So IL-10 can suppress IL-1 and IL-6. Now, one of the most important things that we have gained a recognition of is how we can use these interleukins and measure these as biomarkers of cellular damage. And there's a whole science on how to use this stuff. When we talk about biomarkers, this is as good a time as any to talk about it, but when we talk about biomarkers, we generally talk about cellular biomarkers and we talk about functional biomarkers. When we talk about a cellular biomarker, we're looking for something that specifically tells us about how the cells of a tissue or an organ are actually working. Functional biomarkers tell us how the organ itself is working. So a great example of that, we talked about this in one of the other lectures, but let's go back over it. So I hate going back to the heart. You know how much I like kidneys. But let's look at the heart again. So if we look at a cellular biomarker, we are looking at troponin. If we are looking at a functional biomarker, we're looking at the EKG. The important thing about any biomarker is that we are looking for very high sensitivity as well as specificity. So sensitivity goes to whether or not it picks up the disorder or picks up the cell damage. Specificity of any biomarker has to do with how narrowly released is that biomarker by a specific set of cells. So once again, let's go back to a couple of examples. When we look at sedimentation rate and C-reactive protein, remember we talked about that just a few minutes ago in terms of it being an inflammatory marker. These are inflammatory markers that are fairly sensitive, which means that if you have an inflammatory process going on, sedimentation rate and C-reactive protein are likely to go up. However, they are not specific. The sedimentation rate and an elevated sed rate and an elevated CRP or C-reactive protein only tells you that there's an inflammatory process, sensitive. It doesn't tell you where that inflammatory process is. It doesn't tell you whether it's a rheumatoid arthritis or whether it's a vasculitis or whether it's a glomerulonephritis. So that idea of biomarkers, cellular versus functional, as well as the concept of sensitivity and specificity, very, very important. Two other cytokines are interferons and tumor necrosis factor, or TNF. The primary job of an interferon is protection against viruses as well as to modulate the inflammation and the inflammatory process. What's really cool about interferons is that they're actually released by the cells that are infected by the virus. So when a cell becomes infected by a virus, the cell itself will release the interferons. Now, it's important to understand that interferons are not virucidal, which means they can't kill the virus. However, they can protect the virus from infecting other cells through their antiviral proteins that they produce. Key concept of interferons. Hint, hint. So what's tumor necrosis factor? Usually we just call it TNF, but there are different forms of tumor necrosis factor, the most common of which is alpha, and the one that we know the most about. TNF is secreted by macrophages and the mast cells. And TNF and remember, mast cells have to undergo degranulation in response to injury. Tumor necrosis factor is pro-inflammatory with both local as well as systemic effects. Now, TNF doesn't come without a price, so the positive effects are that it facilitates chemokine production as well as fever. And we know that fever is intended by the organism to be a protective mechanism. Raise the body temperature, make the internal environment uncomfortable for whatever has invaded so it goes away. However, the negative effect of TNF is that if this is prolonged, it leads to cachexia and leads to shock syndromes. So when we think about TNF, we think about balance. A little is useful, but too much can clearly be damaging. Let's take a closer look at some of the individual cells that we've been talking about. We're going to take a closer look at mast cells, and we're going to look at phagocytes. Mast cells are found primarily in our connective tissue, and in those connective tissue, they tend to be pretty close to our blood vessels and in those areas that have a lot of exposure to our environment, which is why you see a lot of mast cells in the lungs as well as the GI tract. Mast cells are activated by a large variety of stimuli, and that's an okay thing because that biggest stimulus is, correct, cell injury. So when we look at that and those stimuli that are leading to cell injury, this could be due to heat, it could be due to a chemical agent, it could be because we've activated the immune system, we have invading bacteria, or viruses acting on receptors. Mast cells release their products through degranulation, and they can also synthesize new mediators. One of the important things that mast cells do when they degranulate is not only stimulate the release of cytokines, leukotrienes, prostaglandins, and our neutrophils, they also stimulate the release of histamine. As we identified, we'll use the royal we, identified there are both H1 and H2 receptors. The role of histamine is to be vasoconstricting in large vessels and vasodilating in the small vessels. And the idea behind histamine then is to increase the microcirculation as well as increase vascular permeability. Mast cells also release those chemotactic factors, the neutrophil and eosinophil factors, as well as stimulate the synthesis of those new mediators, the ones we talked about, leukotrienes, prostaglandins, and platelet activating factor. I hope you find the chart, the diagram that I provided along with these slides to be helpful as you study cellular injury and the inflammatory response. That was developed by Dr. John Gonzalez, and he has my thanks for doing that, believe me. Sorry about that. So what are the phagocytes? When we think about phagocytes, we are thinking about white cells. And so my expectation is that you be able to connect the specific type of phagocyte to what they actually do and what their role is in protecting the organism. So neutrophils are predominant in early inflammation, and like every other phagocyte on the planet, they have that ability to engulf. And so they are especially good at engulfing bacteria, dead cells, and debris. So when you look at an elevated neutrophil count on your white blood cell count, look for a bacterial infection, because you look for this in bacterial response, a response to a bacterial invasion. Eosinophils engulf parasites and also assist in regulating mast cell vascular mediators. Although eosinophils are important in parasite infections, the other place that they're very important with and extremely useful is in allergic responses. So when you see an elevated eosinophil count on your white cell count, think about, is this parasitic or is this an allergic response, and go looking for the allergen. Basophils can also be seen with allergic responses and are a source of IL-1, interleukin 4, not IL-1, interleukin 4. Interleukin 4 is important in B cell as well as antibody production. Monocytes and macrophages are phagocytes that initiate the cellular inflammatory response. And then last but not least, the NK cells, or the natural killer cells. Natural killer cells function to recognize and eliminate viruses as well as some cancer cells. All right, so all of these cells, the neutrophils, eosinophils, basophils, monocytes, and the NK cells, these are all phagocytes. In addition, because they are all phagocytes, they play an important role in activating the acute phase reactants that are made primarily in the liver. So these phagocytes play an important role in activating complement, kinins, and that coagulation cascade. So how do phagocytes work? Well, they work by phagocytosis, but how do they do that? So the ability of a white cell count, the phagocyte, to get to the site is critical, and for it to be able to not only get there, but also to be hungry, to engulf. So phagocytosis occurs when phagocytes leave the circulation and actually enter the site of inflammation. As we know, phagocytes migrate via chemotaxis to the area of highest inflammation. So it's that idea of getting there and then being hungry. In chronic, just to give you an example, in chronic kidney disease, especially in advanced stages of chronic kidney disease, grade 5, we know that leukocytes don't function as well. White cells don't function as well in their phagocytic role. When someone is uremic, advanced kidney disease. Their phagocytes, especially their leukocytes, are slower, so they don't migrate as quickly. So their chemotaxis is decreased, and at the same time, they're not very hungry, so they don't do as good a job in engulfing the offending organism or whatever called them to that area. So in chronic kidney disease, one thing we know about our phagocytes is that they're slow and they're anorexic. Okay, so back to phagocytosis. Once a phagocyte enters an area, once they're there, they begin to trap and engulf. Opsonization is an important first step to actually trigger more of the phagocytosis. Opsonization is the recognition and the ability to adhere to the virus, the bacteria, whatever. The next step is engulfing. And in engulfing, the important job is to be able to ingest and take in. The phagocyte actually has to take in other cellular material. And they do that by myeloperoxidase. Myeloperoxidase is an enzyme that is responsible for much of the killing effect of the phagocytes, and that happens during the engulfing stage. Following that ingestion and surrounding that, they begin to fuse. And when that fusion stage, they actually connect with the lysosome granules of the offending organism. As a result, then, they are able to destroy the target. Also important in phagocytosis, it includes oxygen uptake by the cell in order to generate reactive oxygen species. You know what those are. These are oxygen-dependent killing mechanisms to actually, for example, kill the bacteria. Now, it makes sense that somehow we have to have a way to turn these things off, because if they just kept on running amok inside an area of inflammation, then you wouldn't have any cells left. So the destructive effects of phagocytosis and the phagocytes has to be mediated by serum inhibitors, so to basically turn them off. After the initial very rapid response by our phagocytes, monocytes then arrive later for the long haul. And the monocytes are there for longer-term defense, as well as to keep a good environment and a positive environment that supports wound healing. Take a breath, right? Okay. So how do we know then that there's any evidence of acute inflammation? Well, we have fever, leukocytosis, and back to those plasma proteins. So let's look at fever. In general, fever is a lot of fever, or a major mechanism rather of fever, is the release of IL-1, interleukin-1, by the neutrophils and the macrophages. Remember that interleukins are messengers. Yes. And what happens is that when you have the stimulus of the fever, you have a pyrogen that acts on the hypothalamus to control the temperature. So that's what the pyrogens are doing. Now we have to realize that we have some issues with age ranges with IL-1 as well as the activity of the hypothalamus. Fever in a child can go very high and can be very difficult to control, simply because the hypothalamus and the response to that is immature. This is why when we talked about fluid balance, this is why we have so many a lot of difficulty with fluid issues in a child, because they don't respond well to fever, and they have an immature response, and so they lose more and more fluid because of vasodilation. We also know that our fever response is less sensitive and less responsive in elders. As a result, then, a low-grade temperature is likely to be significant in an elder that in a 40 or a 50-year-old you wouldn't see as significant. In addition, as we get older, our hypothalamus response our response to an IL-1 generated fever in the hypothalamus is decreased. Leukocytosis. Leukocytosis is an elevated leukocyte white cell count. Now, you've probably in practice heard about a left shift. So what do we mean by this? When we talk about a shift to the left, we mean that there are a higher than what we would expect normally percentage of immature cells, especially bands, in the circulation. And this is called the bandemia. It's normal for us to have less than 6% of our white cells as bands. Now, how did this left shift idea come about? Well, when scientists were beginning to understand leukocytes, they were drawing them. And so they were drawing the stages of a leukocyte development. And so when they started their drawing, they started the immature cell on the left, and their drawings of the mature cells went to the right. So the cell drawing on the left was the most immature. The one on the right was the most mature of the white blood cell. The other thing is a band is an immature white cell that can pretty much become almost anything. Now, the importance of a bandemia, or a higher than 6% level of bands in the white cell count, is it's telling you the body is trying to respond to the infection. So in many ways, you take that as a good sign because it means that the organism is responding appropriately. So what would the bad part about a bandemia be? Shout at the screen. Yeah. They're immature, and so as a result, they cannot do the job that a mature white blood cell could do. So there is both a positive as well as a negative. So then our third piece of the systemic evidence of acute inflammation are the synthesis of those plasma proteins. We've already talked about this, so we can talk about this as the liver producing those acute phase reactants. Now, the ones we talked about so far was C-reactive protein. However, there are other acute phase reactants that can signal inflammation, and those include fibrinogen and ferritin. Fibrinogen we think of in terms of our clotting mechanism, right? And in ferritin, we think about that in terms of our iron storage and our iron use mechanism. However, it's important to understand that C-reactive protein, fibrinogen, and ferritin also increase in acute inflammation, and you have to incorporate that in your thinking as it could be an inflammatory response as opposed to something going on in a clotting mechanism or in an anemia. I know this has been a lot of information, and this is where there's so much crossover between normal physiology but recognizing how that can lead to pathophysiology. And we can't understand pathophysiology if we don't understand the normal. And we are going to talk so much about inflammation and the response and the inflammatory response and the responses of our immune system all the way through the course that I think it's important to have a good understanding of it. We know that inflammation starts with cell injury and mast cells. Remember that degranulation of the mast cells so that their products can be released to the circulation. The inflammatory response has a lot of components that have to work together to respond to that injury. And these components are both homeostasis supporting as well as potentially pathologic. Phagocytes and phagocytosis are key players in inflammation, so we need to know which ones are responsible for what and what we would look for, as well as the steps that they take in order to do their job. Because when we understand how we respond to injury, we can then understand how and why many pathologies develop. I hope you have found this helpful. In addition to your reading, focus on the things that we've talked about. Remember, I will not test you on anything that we have not discussst, but there are some real, not only good pictures, it's not Playboy, but there are some good pictures in McCants and Huther, as well as an additional level of explanation. Thank you so much for your attention, and again, let me know if you have questions.